Assalamualaikum and hi. Today me and my friend will be presenting about a topic that connect chemistry with our everyday life, which is about artificial coloring in beverage by using the chromatography paper. First of all, what is artificial coloring? Artificial colors are a man-made food color or color additive. They are often added in food beverage to enhance its appearance, making it more appealing, colorful, and even attractive to the consumer. There are a 7 types of common dye that are usually approved and certified by food authorities. These include the color blue 1, blue 2, green 3, red 3, red 40, yellow 5, and even yellow 6. These colors are widely used in food products like candies, drinks, and even snacks. Now, let's move on to the next part, which is why is it important to detect artificial coloring. There are several important reasons, which is the first one is health concern. Some artificial color may cause some allergic reaction or health issue, especially to the children. That's why detecting and controlling them is important to protect public health. The second one is regulatory compliance. Food industries must follow the rules set by health authorities. By detecting artificial coloring, it helps ensure that food products meet legal standard and safety regulation. Lastly is quality control. By monitoring the use of artificial color, manufacturers can maintain consistent product quality and avoid contamination or even a mistake. Let's look at why we use chromatography paper to detect artificial coloring. There are a few reasons why we use chromatography paper. The first reason is because of visual identification. Chromatography paper helps us see the different dye. Each dye travels at a different rate depending on how soluble it is in the solvent and how it interacts with the paper. So, just by looking at the paper, we can identify which dye are present based on how far they travel. The second one is separation of components. If a drink contains a mix of different dyes, chromatography helps to separate them. Instead of just seeing one combination of color, we can break it down and see all different dyes that make up to the color. And the last reason is because it cost effective and simple. One of the best reasons why I use the chromatography paper is because it's cheap and easy to use. We don't need any advanced lab equipment like the beaker, pipette, and others, but just some paper, solvent, and a sample to use the chromatography paper. That is why it's perfect for school, lab, and even a quick food safety check. Overall, chromatography paper is a practical and powerful method for analyzing artificial coloring. Here are some types of common drinks that often contain artificial coloring, like the fruit flavored juice, carbonate soft drink, yogurt and flavored milk, and even the bubble tea.
First of all, let's know what is chromatography. Chromatography is a separate component of a mixture by disturbing them between a stationary phase and a mobile phase. The components travel at a different rate based on their differing interactions with these phases that leading to separations. So what is stationary phase? The stationary phase in chromatography is a solid or a liquid coated solid that stays fixed inside the column while the mobile phase moves over it. It enables the separations of compounds based on their interactions with the mobile phase. However, mobile phase is a liquid or gas that carries the sample through the chromatography system, allowing its component to separate based on their interactions with the stationary phase. This is an overview of chromatography paper to help us more understand the comparison between stationary phase and mobile phase. Now, look at the right beaker. We can see the stationary phase that started in the place. Stationary phase will be absorbed by the components that are being separated where the solvent and analyte or components travel through it. Meanwhile, the mobile phase at the bottom, which is the elementing fluid that may be water or alcohol, will be moved over and through the stationary phase, carrying with it the analyte mixture. So, the left beaker is a result of separations of sample spot after this separation process occur. As we can see, the components separate by their category of color like a picture in the slide. Next, we may ask how the separation process occurs, right? So firstly, the components that are strongly retained or observed by the stationary phase will be moved slowly with the flow of mobile phase. So, components that are weakly held by the stationary phase will travel fast, which is rapid, with the flow of mobile phase. After that, the differences in mobility for each component in a sample will be separated out into discrete bands. Then, it can be analyzed qualitatively and quantitatively.
Okay, next I will explain the methodology used to detect artificial coloring in 4 selected drinks using chromatography paper. First, Draw a straight line with a pencil 3 cm from the bottom of the chromatography paper and mark 4 small X along the line, each about 3 cm apart. Then label each X with the drink names, which is strawberry syrup drinks, lemon syrups, blue raspberry cordial, and green apple soda. After that, use a capillary tube to place a small drop of each drink on its label X. Repeat the spot if the color is too light. Next, prepare the solvent by mixing 2 grams of NaCl with 100 ml of water. Pour about 50 ml of this solution into a clean beaker. The next steps, hang the chromatography paper vertically using a clamp and stand. Dip the bottom of the paper into the solvent, but make sure the pencil line stay above the liquid level. After that, let the solvent rise halfway up the paper. Once it reaches the middle, remove the paper and quickly mark the highest point of the solvent reaches, or also called the solvent front. Last but not least, leave the paper to dry for 5 to 10 minutes. Once dry, observe the separated color bands formed by each drink.
This is the example result of a chromatography test to show how artificial colorings in drinks can be separated and studied. The example includes 4 different drinks: strawberry syrup, lemon syrup, blue raspberry cordial, and green apple soda. In the chromatogram, each sample showed between 2 to 4 separated color bands, which indicates that these drinks contain a mixture of dyes, not just one. Among them, the green apple soda showed the most color bands, meaning it likely contains the highest number of artificial colorings. To analyze the movement of the dyes, we use the RF value, which is calculated by dividing the distance the dye travels by the distance the solvent front moves. For example, if a dye moves 3 cm and the solvent moves 10 cm, the RF value is 0.3. Dyes with low RF values are more polar because they stick to the paper and move slowly. Dyes with high RF value are more non-polar because they travel further with the solvent. Looking at the example result, the strawberry syrup separated into 3 red sheets that stay near the bottom of the paper, showing that it contains polar dyes. The lemon syrup had 2 yellow tones that move a medium distance, which suggests moderate polarity. The blue raspberry cordial showed 3 color bands: dark blue, light blue, and a hint of purple that travel higher up the paper, meaning those dyes are mostly non-polar. Finally, the green apple soda showed both low and high bands, including yellow, green, turquoise, and blue, which means it contains a mix of polar and non-polar dyes. These results show how chromatography can help us not only detect how many dyes are in a drink, but also help us understand the chemical behavior of those dyes based on how far they move.
In conclusion, the chromatography paper test will successfully reveal the different artificial colorings present in each beverage. By analyzing the number and position of color bands, we could determine not only how many dyes we use, but also their polarity based on how far they can travel. For example, the green apple soda showed the most color band, which is it indicating the contain the greatest variety of dye, which is contain both polar and non-polar. On the other hand, drinks like lemon syrup and strawberry syrup had a few bands, suggesting it has a simpler dye composition. This experiment highlights how important it is to detect artificial coloring in food and drinks. It supports health safety, ensure compliance with regulation, and maintain quality control. Overall, chromatography proved to be a simple, effective, and visual method for identifying synthetic dye in beverage.